Nucleic acids, DNA, RNA, and genetic information. Nucleic acids are some of the most important molecules in biology. The two primary types of nucleic acids are DNA and RNA, which are essential to the storage and transmission of genetic information. Understanding these molecules and their roles in biology is critical to understanding the fundamental processes of life. DNA, or deoxyribonucleic acid, is the most well-known nucleic acid. Its structure was first described in 1953 by Watson and Crick, who showed that it has a double helix structure. The structure of DNA is made up of nucleotides, which are composed of a phosphate group, a sugar molecule, and a nitrogenous base. The four nitrogenous bases in DNA are adenine, A, guanine, G, thymine, T, and cytosine, C. The sequence of these nitrogenous bases is what encodes genetic information. The information is stored in a linear sequence of bases along the DNA molecule. The order of these nitrogenous bases determines the structure and function of every living organism. For example, humans have about 3 billion base pairs of DNA, which determine their genetic code. DNA replication is a crucial aspect of life. It is the process of making a copy of all of the genetic information in a cell. DNA replication occurs when the two strands of the double helix are separated, and each strand is used as a template for the creation of a new complementary strand. The end result is two identical DNA molecules. This process occurs during cell division, so that each new cell has a complete set of genetic information. RNA, or ribonucleic acid, is another important nucleic acid. RNA differs from DNA in a few key ways. First, it has a single strand structure, as opposed to the double helix in DNA. Secondly, RNA uses the nitrogenous base uracil, U, instead of thymine, T. Finally, the sugar molecule in RNA is ribose, instead of deoxyribose in DNA. RNA has several different types, including messenger RNA, mRNA, transfer RNA, tRNA, and ribosomal RNA, RNA. mRNA is responsible for carrying genetic information from DNA to the ribosome, where it is translated into a protein. tRNA serves as an adapter molecule, bringing amino acids to the ribosome where they are linked together to form a protein. Finally, RNA forms the structure of the ribosome, where the process of protein synthesis occurs. The process of transcription is how a DNA sequence is used to create an RNA molecule. This process occurs in the nucleus of eukaryotic cells, where the DNA is located. The DNA strand is used as a template to create a complementary RNA strand. This RNA strand, known as the primary transcript, is then processed to create the final mRNA molecule. The mRNA molecule then leaves the nucleus and travels to the cytoplasm, where it is translated to create a protein. The genetic code is the set of rules by which information encoded in genetic material is translated into proteins. The code is composed of codons, which are groups of 3 nucleotides that specify a particular amino acid. There are 64 possible codons, but only 20 amino acids that are used to make proteins. This means that multiple codons can code for the same amino acid. The genetic code is nearly universal, meaning that it is used by almost all organisms on Earth. This has led scientists to believe that all life on Earth shares a common ancestor. Mutations are changes in the sequence of DNA or RNA that can lead to genetic variation. Mutations can occur spontaneously or can be caused by radiation, chemicals, or viruses. Mutations can be harmful, beneficial, or neutral, with the majority of mutations being neutral. Harmful mutations can lead to genetic disorders, while beneficial mutations can help organisms adapt to changing environments. Modern genetics is the study of the function and behavior of genes. Genetics is a field that has seen tremendous progress in recent years, thanks in large part to advances in technology. The study of genetics has led to new treatments for diseases, such as gene therapy. Gene therapy is the use of genetic engineering to treat or prevent disease by modifying the genome of a patient. The field of genomics is the study of the complete set of genes in an organism. Genomics has led to the sequencing of the human genome, which was completed in 2003. The human genome is composed of about 3 billion base pairs of DNA. The sequencing of the human genome has led to new insights into the biology of humans, as well as advancements in the treatment of genetic diseases. Additionally, the study of genetics has also shed light on the evolutionary history of life on Earth and the mechanisms by which organisms adapt to their environments. The genetic code, which is nearly universal, has allowed scientists to trace the ancestry of organisms and the relationships between different species. However, genetics also raises important ethical considerations. The ability to manipulate the genome raises questions about the moral implications of altering the genetic makeup of humans and other organisms. Furthermore, genetic testing raises concerns about privacy, discrimination, and access to healthcare. It is important to note that genetics is a constantly evolving field, with new discoveries being made all the time. As technology advances, so too does our ability to study and manipulate genes. In addition, genetics has also contributed to our understanding of complex diseases such as cancer. The study of cancer genetics has led to the development of targeted therapies, which can be tailored to the specific genetic mutations present in a patient's tumor. This approach has shown tremendous promise in improving patient outcomes and could represent a significant advance in the fight against cancer. The future of genetics is promising, with the potential for even more groundbreaking discoveries and advancements in the years to come. Scientists are actively working to develop new tools and technologies that will allow them to better study and manipulate genes. One area of particular interest is epigenetics, which studies the changes in gene expression that occur without changes to the underlying DNA sequence. Epigenetic changes have been linked to a variety of diseases, including cancer and neurological disorders, and understanding them could lead to new treatments. Another area of interest in genetics is synthetic biology, which involves the design and construction of new biological parts, devices, and systems that don't occur naturally. Synthetic biology has the potential to help us address a variety of challenges facing our society, including climate change, energy production, and disease prevention. As genetics continues to advance, it will be critical to ensure that its benefits are accessible to all people, regardless of their socioeconomic status or geographic location. The democratization of genetic technologies, such as genome sequencing and gene editing, will be essential in ensuring that everyone has access to the benefits of these advancements. With new tools and technologies, scientists are working towards a comprehensive understanding of nucleic acids in genetics. The role of epigenetics and synthetic biology in advancing this field cannot be overlooked. Epigenetics has helped us understand the important changes in gene expression that occur without the presence of mutations in the DNA sequence. Synthetic biology has the potential to explore new possibilities in energy production and disease prevention. Moreover, genetics has a significant impact on society, and it is vital to ensure that everyone has access to its benefits. It is necessary to democratize genetic technologies and make them accessible to all, irrespective of their location and income. By doing so, we can ensure that everyone benefits from the advancements in genetics, and the benefits of genetic therapies are not restricted to a select few. Genetics has also impacted fields beyond medicine and biology. It has implications for fields such as agriculture, forensics, and even anthropology. The study of genetics allows us to understand how organisms evolve and adapt to their environments, which is particularly useful in agriculture. For example, modern agriculture relies heavily on genetically modified crops, which have been engineered to resist pests and herbicides.